Okay, now I'm going to try to explain a little bit more how nucleoprotein allows the negative stranded RNA viruses replicate, makes an antigenome, a positive strand antigenome that is of the exact length of its original negative stranded genome. Okay, so remember last time in the previous slide, I explained that for negative stranded RNA viruses such as influenza virus, it produces a positive stranded RNA strand which function as the mRNA, but these strands are shorter, and thus the virus cannot use this type of strand to use it as a template to make its genome. So it has to make something that is the full length of it. Okay, in this picture that I'm showing you up here, that is how the negative stranded RNA virus RNA polymerase looks like. So you have those in 4 different views, the front views until the back views. What you wanted to look at it is actually from the back views. It looks like your palm and the fingers. And in this image, what I want you to look at this is at the same orientation. On the left side, you have the palm and the fingers facing the fingers on the top, the palm at the bottom. So it is at the same orientation as the image above. But what you want to focus is on the left image, and it pointed there that look at how the mRNA that is produced. It is when the mRNA is produced, or whatever positive stranded are being produced. So you have the yellow strand that is the genome, and then the growing strand or the positive strand. When it is produced, it is actually threaded between the palms and the fingers. So you can imagine that when the polymerase moves along the negative strand genome, it will drag the mRNA or the positive strand along with it. Okay, so I'm not going to manually draw a line there. So imagine that now the positive strand is growing longer, and because now you have a lot of nucleoprotein in the cells, the nucleoprotein will bind on these positive stranded RNA. So you bind and cover all these mRNA. Remember earlier also that we explained about how the polymerase, when it's reached the stop signals at the end of the negative stranded genome, it will do this process that we call stuttering. That means it will stop at that particular uracil. Instead of going forward, it keeps going back and forward multiple times, about 100 to 20 times, until and each time you will add new adenine to it, so until you manage to get lots of long poly A tail. So remember that stuttering process. Now imagine that you have this nucleoprotein already binding to this positive stranded antigenome. Because that nucleoprotein is there, there's no way that the polymerase can backtrack anymore. Okay, so you have that and then nucleoprotein binding to it. The only way that the polymerase can move is moving forward. So this is what this is explained. This is what we are trying to say, or explain why only when you have enough nucleoprotein to cover the newly synthesized positive stranded RNA, it will prevent this stuttering effect. And eventually what happens is the polymerase have to, the only way they can move is just straight on ahead and finish reading to the whole length of the negative stranded genome. And finally what you get is the positive stranded antigenome, which is the exact length of this negative stranded genome. Now these situations that we are explaining right now happens in all negative stranded RNA viruses, influenza virus, Ebola, hepatitis C virus, all that. The only difference for influenza virus is its RNA polymerase is not capable of making its own cap structure. That's the only difference. The rest of this situation that we're explaining right now, including the formation of poly A tails, that is applicable to all negative stranded viruses.